This fall, we started to work with Don Avers, the waterfowl biologist with Michigan DNR, to sample waterfowl that congregate at Quarry Marsh Ecological Research Center during fall migration, and we were sampling them for avian influenza virus. We just initiated this research, so we don't actually have a lot to share with you today, but we can at least provide you the rationale for why we're doing this and the methods that we use to sample ducks for influenza. So what is avian influenza virus? And is that related to the influenza viruses circulating in humans? Does it relate to the seasonal flu that we're all familiar with? Well, yes and no. Avian influenza virus is a type A virus, influenza virus, which is just one of 3 types of influenza viruses that circulate in human populations. Now, while most influenza A viruses do originate in wild birds, particularly waterfowl, dabbling ducks and geese, most do not pose any risk to human health. However, there are some types of avian influenza viruses that can be particularly devastating for domestic poultry, including chickens and turkeys. Avian influenza viruses are divided into 2 categories. They're classified as low pathogenic and highly pathogenic. Low pathogenic is what we mostly detect in wild birds, and birds infected with this type typically show mild symptoms or remain asymptomatic. Highly pathogenic avian influenza, which is rare, evolves from certain subtypes of low pathogenic strains. Now, while they do originate in wild birds, they typically become virulent within domestic poultry. However, there are instances of wild birds being the direct source of a highly pathogenic avian influenza virus outbreak in domestic poultry. Highly pathogenic strains of influenza virus can cause up to 100% mortality within 48 hours in domestic poultry. Outbreaks of high pathogenic avian influenza virus are rare, but we did have one in the United States, or have multiple in the United States between December 2014 and June 2015. During that time, multiple subtypes of high path influenza were detected in backyard flocks and commercial poultry operations in 21 states. In total, the virus impacted over 20 backyard flocks and over 200 commercial operations. And over 50 million birds were euthanized to prevent the further spread of the virus. So the economic toll of high-path avian influenza virus is substantial. Now, we never know, like most viruses, when the next high-path will emerge. And surveillance efforts are important to detect potential threats before they cause an outbreak, so that control and prevention strategies can be implemented. A long-term surveillance program will allow us to see patterns in prevalence between years and the types of viruses circulating in wild bird populations. We also see this as a great opportunity to provide training for students and scientists in sampling techniques. Given the role of migrating waterfowl in the potential dispersal and movement of these viruses, and that the juveniles, those first-year birds, are the most likely to be actively infected, we see most of the circulation among waterfowl in the late summer, early fall. Therefore, our sampling efforts are concentrated in the fall months, which is nice because it aligns with the state's duck banding program. Now, we sample for influenza virus just in general, and if we detect samples that have potential to be a high pathogenic strain, because there's only certain subtypes that have potential to be highly pathogenic, if we detect those, then further testing would be done. It would be very rare to find these in ducks, but you would never know unless you looked. So here's a picture of actually how we capture the birds. We use some swim-in traps. And in those traps, they're baited and they're open, so ducks can get comfortable. They move in and out for the food for a a couple weeks, and then one day they'll get set so when the ducks move in, they won't be able to get out. So this is what that can look like at Quarry Marsh. You can see quite a few wood ducks and the swim-in trap. Then the next day after setting the trap, you go back and you capture the birds that are inside the trap. And this is how they capture them to ban them, and also how we capture them to process them for influenza virus. And I wanted to point out just the two most common species that we catch, as shown on previous slides, are mallards and wood ducks, which are very abundant in the fall. So these are primarily the species we would be collecting samples from. So this is a fecal oral transmitted virus. So when a bird gets infected, the virus typically will bind into the lower intestinal tract or upper respiratory tract. And we collect swabs either from the mouth or from the cloaca, and then we place that in a solution so that we can assay for the virus at a later date. Here we just have pictures demonstrating what that would look like, but not the actual sampling. So where do we go from here? Well, this time we just have begun sampling efforts and we have no results yet. But hopefully in future field days, virtual or in person, we can share our findings. Thank you.